And thanks for the organizer. This has been till now and will be an exciting workshop and with so many questions. And I'm learning a lot, actually. So, yes, I'll tell you the story of why I got interested some time ago in trying to understanding more about cell extracellular matrix interaction. My interests are essentially in development of tumors, tumor growth, cancer modeling, and tissue remodeling. And in many of these problems, one has to face the problem that the tumors that is growing, for instance, interfacing some structures, I don't know, the basal membrane, for instance, or in any case, it's interacting with the extracellular matrix very strongly, both as a confinement measure and in order to crawl or invade, and so the other interest in invasion and metastasis. Okay, so this is the final end, and at some point, and this is will cover the first part of the first half of my lecture. I saw these movies by Peter Frieder and Katrina Wolf, in which you have in the same collagen two types of cells behaving completely in a different way. Okay, so this is a cell in a collagen, and you see that there are long protrusions in the 3D extracellular matrix, but the nucleus, the yellow stuff over there, is stuck and cannot go through. Okay, and will stay there more or less forever. Well, of course, here in this cell, for sake of completeness, metalloproteinases, that is the action. I'm telling that for the students, the action of the cell that is diffused to break up the extracellular matrix is completely inhibited. Okay, instead this is in the same collagen with the same density, the behavior of neutrophil, and you can see how instead it can squeeze through the net of the extracellular matrix. So this, of course, has to do with one single cell. As I told you, I'm more interested in describing ensembles of cells, so many cells, from the macroscopic point of view, using continuum mechanics in some sense. And the aim here was then, it has been then, and will be in this talk, to try to merge this microscopic information that you get by looking at single cells, or what happens at the subcellular level also, and bring that information to the macroscopic scale. Whether this is a multiscale modeling, I don't know. I mean, a multiscale upscaling, I'm not really sure, and you will see why. Okay, so the basic stuff, I will not enter into this equation too much. The basic stuff for describing tumor growth from the macroscopic point of view is to write a mass balance equation, which is the first equation over there, and possibly some force balance equation. And the key ingredients there are, of course, the right-hand side of the first equation that describes growth, and in the second equation you have the stress, how cells behave, how cells interact, and the crucial one that I will be focused on today is the other term, this M, that is the interaction force between the cell and the extracellular matrix. And in most of the paper, this term is taken to be proportional to the velocity. And we've already seen some example of that, for instance, in Eva Paluk's talk, for instance, and others. Okay, this is things that one can do. I mean, this force is just proportional to the relative velocity of the cell with respect to the extracellular matrix, like a viscous track. Okay. Well, the aim here then, the plan of this lecture is that instead to see in a little bit, with a little bit more in detail, why and how. Instead, this term should depend on many microscopic features, namely the pore size, the nucleus size, the elasticity of the nucleus, the elasticity of the fibers, and then why the behavior of this ensemble of cells have a bimodal behavior, and bimodal behavior. And this is true in several aspects. It is not only in one single feature. Okay, but keep in mind that though the talk is devoted to look at what happens at the cellular scale, my interest is always to bring that information up. Okay? Okay. So these are, starting from the experiments, these are the experiments by Katerina Wolf and Peter Friedel. You see here an aggregate over there that is growing in a gel, in a rat tail collagen, and it's invading in 18 hours. Okay, this is the same, well, not the same, but similar aggregate that is actually not growing in a gel. Okay, you see those spikes over there, but I show you the movie of one single cell. These are not cells that are invading the gel, but just protrusion of the cell, extension of the cytosol into the gel, but the nucleus is stuck at the border of this aggregate. Now, what is the difference between these two? The difference is the following, that the former gel was produced at a lower temperature, and is this one over here, this one. Okay, and you can see that is the pore size are much wider than the one above. More or less, the cross section is of the order of 10 microns square. Instead, the second one is obtained at 37 degrees, and is this one. Okay, the density is the same. The density of the collagen is the same. So the density is not a feature. Okay, it's the microstructure that brings a huge difference between invading or not invading. Again, here metalloproteinases is inhibited.
A question?
Yeah.
So if the density is the same, and the thickness of the fiber is much larger.
So here you see fibers that are much larger than this one, and so it means that, say, the volume occupied by the fiber is distributed differently, so that this thickness is more. And so the key feature is the pore size of the network, that, as you can imagine, has to be compared with the pore size of the nucleus, okay, at the end of the story. Okay, this is very similar in the sense that on the right you have the rat tail collagen, and on the left a bovine collagen. Bovine collagen have a much thicker fibrils, and so with the same density again, same density, you have different behavior. Okay, and I'm showing you this movie. These are just, okay, in this first part, let's see, yeah, in this first part, I hope you can see, yes, you can. Okay, this is bovine, this is rat tail, and they are both moving because of the action of metalloproteinases. So the cells are able to cut the fibers and to build the road to advance or to move in a, say, random-like fashion or whatever. Instead, now you will see what happens when metalloproteinases is inhibited by this protein, and you see that the nucleus is there. I mean, it will not move. This is barely moving, more or less, but they are not moving at all. Okay. Well, I had also a better picture, but I forgot. Okay, and this instead is the neutrophil moving, non-moving, the cell not moving or moving according to the collagen concentration. If you go down with the collagen concentration, this will stay there or will just move. The final graph that, I mean, from my point of view is interesting from my application is this one. You have a plot of the speed of the cell. Speed of cells is a function of the cross section, and from this you can infer that there is a threshold value, because below this cross section cells are not moving, and then they are moving more or less, I would say, in a linear fashion. I don't know. Okay, so how can we build on that some modeling? Of course, the network is very complicated, so let's simplify that. And instead of assuming that this is a mesh, assume that we have channels of ECM or microchannels, and the cell has to pass through this microchannel. In order to pass through this microchannel, as you can see here, they have to squeeze through these pores and go through. Okay, the cytoplasm is more or less, it's very easy for the cytoplasm to advance, but not for the nucleus because it's the stiffest part of the cell. And we did a lot of, say, trials, in the sense that, of course, you can assume a lot of things on the deformation of this nucleus. Yeah.
You have these fibers. There's a lot of space between the fibers. Is it the case that the cell goes through the fibers, or sorry, does it have to make its way through the fibers, or did the fibers become absorbed in the cell? Basically.
Sorry, I didn't understand the end of the
question. There are fibers absolutely everywhere.
Yeah.
But there's still a lot of space.
Mm-hmm.
My question is, does the cell make its way in the gaps between the fibers when it moves?
If it's using metalloproteinases, it's opening the gap.
It's moving in the gap.
Yeah, it's opening the channel in it's making channels actually. Okay.
Digests the fibers.
Oh, it can digest the fibers.
Yeah, they can cleave the fibers.
Oh, it eats the fibers.
Cleave the fibers and digest the fibers.
Dissolves.
Okay, so so we are assuming that, for instance, this nucleus deforms as an ellipsoid or in a cigar-like shape, but this doesn't make any difference. And the other important part is what? Is the traction forces, of course. In order to advance into the extracellular matrix, the cell has to exert forces on the ECM, on the extracellular matrix, and pull it into the network. Of course, these are two-dimensional pictures. Okay.
Sorry, I'm curious about what you assume for the mechanical properties of the nucleus, because dishes group and so on. There have been quite a bit of discussions how one should model it.
Okay, it's not that easy, and in some sense we're waiting for some measurements. At the moment we are assuming that it is elastic with a membrane. So it's an elastic body inside with a membrane, and by the way, is preserving volume, which is also not true. Okay, but we have to start somewhere in some sense. So that's the minimum. Of course, here, as we will see, the computations are in a quasi-static way, so viscoelasticity is ruled out. If you want, I can say this is viscoelastic, but it doesn't make any difference. It's elastic. Okay, so this is the picture of the forces. I might come back to that later on if I have time. This is a picture done by traction force microscopy, and you all know that cells pull on the front. And what I want to say is that more or less most of the traction is limited to, say, to the tip part of the cell. Okay, and also here we made a lot of assumption, assuming that the force here, the traction force, is linear with the distance from the nucleus, or is bounded to some apical parts of this long, say, protrusion of the cytoplasm that is here, for instance, and so on. Okay, so the main, without entering into details, the main assumption is the following. Well, in order to squeeze the nucleus inside this microchannel, the cell has to do some work. Okay, and the work that needs to be done by the traction forces need to be larger by than the energy required to squeeze the cell. Okay, as simple as that. This is a necessary condition. It's not sufficient, because in principle you could have, say, some bottleneck on the force, not on the work. But this is it. And if you play with a little bit of continuum mechanics, what you get is, according to the assumption that you make, on the nucleus and on the force, you get some, some criterion that tells you whether the cell will go through this microchannel or not. Okay, so let's look at the one at the bottom. This is elastic nucleus, for instance, and if the nucleus deforms in an ellipsoid-like shape, and this is what you get. What does it tell you? Well, it tells you, first of all, that there is a number, a dimensionless number, that I call the Giverso number because she's the one that did the work, more or less. And this number here measure, say, quantities that are related to the traction forces exerted by the cells. That is how strong the cell pulls on the extracellular matrix, the density of bonds that are expressed on the cell membrane, say also the fraction of the microchannel that is functionalized, that is where the bonds can attach. These terms all have to do with how strong efficiently the cell can track, can exert forces on the wall of the microchannel, and on the denominator you have the nucleus stiffness. So in some sense, this number is large if mu is small or if the force is large. Okay? And there is a balance. And, yeah?
What is the unit of this number?
Sorry, what is?
The unit of the number, what?
This is dimensionless. This is a dimensionless number. Okay?
A question?
Yep.
Stiffness, do you mean the Young's modulus or what?
The Young modulus. Okay, so the criterion is this one. This number, okay, if this number is larger than something that is related completely to the geometry of the channel, you see here RP is the radius of the pores with respect to the radius of the nucleus, then the cell is able to go through that network, otherwise it will not. Okay? So it is hard to catch the meaning of that from the formula, but let's look at the graph. For instance, this one. Okay, this is what you have. You have that there is a relationship between, say, geometric characteristics and mechanical characteristics, so that in this region there is no way the cell can go into the channel. Which means that if you have, say, some given mechanical characteristics for the cells, forces given, stiffness, Young modulus is given, then in order, the cell can go through the channel only if the radius of the pore is large enough. Okay, it's large enough. Otherwise, it will not go through. Or, vice versa, given the ECM, given the structure of the ECM, then cells with low G will not go through, and in order to pass through they have to increase G, which means increase the force or lower the stiffness. Or if you have more clones, yes, if you have more clones, the clones that have mu large, stiffer clones, will stay trapped into the ECM, otherwise they will be able to move. Yes, Phil?
What's the origin of this force, this traction force? What's exerting this force on the nucleus?
What? What is this for?
F is the force, right?
Yeah.
Where's that from?
Myosin, and it's an internal force, and it's exerted to the on the ECM by integrins.
But how does that get onto the nucleus?
Well, this is dragging the nucleus, in some sense as usual. It is through the network to the actin network. And so it's dragging the cell in.
Okay.
Maybe if you show it on the picture, just where the force directed.
Well, I don't know if I have a picture.
No, just on the schematic of the channel and the nuclei.
Yeah. Okay, something like
Yeah,
that.
yeah.
Okay. So the forces are acting here on the wall, the channel walls, and I think that the most important assumption is this one. It's here and on this apical part of the cell. Okay, and of course, because of the actin network, it will be in some sense connected to that, and we pull the cell.
It's equivalent to a force that you would apply from outside and dragging it from that region, right?
Yeah.
Yes.
Yeah.
But it's generated by the cell.
Okay. Okay, this tells you something about this threshold value. Doesn't tell anything on, say, the speed at which the cell will pass through the microchannel. In order to to have some hints on that, we use a cellular POTS model, and so in this device, virtual device, you have three microchannels. One is larger than the size of the cell, which is 20 microns. One is smaller than the size of the cell, but larger than the size of the nucleus, which is 10 microns. And one is smaller than the size of the nucleus. Okay? So cells then tend to pass through the microchannel. There is no chemotaxis here, but there is persistence. And so when they reach the microchannel, they tend to move in this direction. And you see that the first two have no difficulties in passing through, even when the nucleus is rigid, but this one is not coming through because the nucleus is too stiff, more or less like this one, this cell here, that is not able to exert forces, as I was told, on the size of the microchannel, though the channel is not too small compared to the size of the cell. It will not go through.
Are these experiments, or these are your results from the model?
This is the experiment, and this is the results of the cellular POTS model. Yep.
Could you image the nucleus in the experiment? So does it really get deformed?
Well, I don't know, because I'm not doing experiments, but I think that you can. And in several experiments, you see that the formation of the nucleus is quite strong. You saw that in the movie by. Let's see if I can go back.
What I mean is, it doesn't depolymerize the shell around of lamin.
No.
It
Not at all.
It does sometimes.
Yeah.
Like in experiments of Mathieu Piel, that's what happens sometimes.
Yes, general question.
There was
Which are the properties of the cytoplasm in relating with the nucleus?
Well, the cytoplasm here is treated as, I mean, it's very easy to be deformed. So actually, it doesn't do any work in order to pass through this very small microchannel. In some sense, it's negligible. It's neglected.
What's the goal of those simulations? Are you trying only reproduce the behavior, sort of, or can you learn anything?
Yeah. Okay. Next slide.
Okay.
More or less. So I wanted to go back to the original, for instance, to this movie here, to show the nucleus. Okay. The red is the nucleus. Okay, for instance, here. In this one, you see this part here, black, is the nucleus. Okay. So, instead, if we have a deformable nucleus, then the cell can go through this microchannel, as in this other movie by Gook.
But in your model, do you treat the nucleus as a separate entity from the cytoplasm?
Yeah, it's another element of the cellular POTS model with its own characteristics, mechanical characteristics. Okay, and you can see here this cell that is squeezing through the microchannel. Okay, so coming to what we can learn on that. Okay, so we can do some graphs of the average velocity that the cell has in this microchannel compared to the pore size. Okay, and there is a threshold, and then it is increasing, and then you have this bimodal behavior. Okay, there is a maximum. The range of the experiments by Wolf are limited to this area here, because of course these are, say, you can achieve these pore sizes probably only with artificial scaffolds, and you can see that here the role of the nucleus is very small, negligible. It's only it has some relevance here, where you have, say, the elasticity more or less of the nucleus that gives some information. And the reason because of that, of course, is that in large pore sizes, it's harder for the cells to find a way to pool, and instead there is a maximum, there is an optimum pore size that is related more or less to the size of the nucleus, so that the nucleus has, say, no difficulties in passing through. The cell is, in some sense, is elongated, and can exert more the tractions on the walls. I mean, this is my explanation.
In your simulation, what's the size of the nucleus that you
It's 10 microns here.
10 microns in diameter.
Yeah.
Okay.
So it's in between here.
Okay.
In between. Okay, good. So we can now go to a structured ECM, like a regular lattice, and again you find the same bimodal behavior of the average velocity. Okay, small pore sizes, you don't have any motion, not optimal velocity, and then again a decay in this case, and changing instead the fiber elasticity will increase the velocity, because of course the cell, even without cutting, can separate and widen the pore. For instance, in those square lattice, of course, the cell can round the squares and then pass more easily, and you can see that from this large enlarging of this dark red area here. But again, you have, say, minimum, maximum, minimum, according to the fiber rigidity, because if it is, the fibers are too floppy, then there is no way the cell can exert traction on these, say, loose ropes, in some sense. Okay. And just to end, this is an experiment that is done in 2D by Politecnico et al. That show this bimodal behavior, which is similar to this one, qualitatively similar to that one. But most of what I will say is in 3D. Okay, so let's go back now to the continuous model and to see how we can, say, use this information in the continuous model. So the first and the third equation is what I already mentioned. Here we are adding an equation for the extracellular matrix that is rigid. This one, this is the density of the extracellular matrix, is rigid, but it can be remodeled. Remodeled by the metalloproteinases produced by the cell at some point. Okay. So, as I told you, this is in general taken to be proportional to the velocity, but we learned that it depends on the pore size, the nucleus size, nucleus elasticity, fiber elasticity, bimodal behavior, and so on. In particular, looking at the pore size and nucleus size, there is an easy way to say, insert this information in this model that tells you that the speed is not just proportional to the force, but that there is a threshold value. There is a threshold value for the pore radius, and then, for instance, this grows linearly. So this means that the velocity is, say, proportional, and then there is this, the pore size of the ECM minus the pore size, the critical pore size. This is the positive part, so that there is no motion when A0 is larger than this AM, and then this is the force that is exerted by the cell.
So this graph looks like it's a fraction of the graph cut or
Yeah.
what happened after.
Probably it might go like this. This is what we are inferring. I'm not really sure.
But in this data, why the graph is cut?
I think that this is the maximum pore size that they can get as a collagen. I'm not really sure. Or at least this is the maximum that they tried as densities of collagen. The interesting part would be to go to artificial scaffolds and prolong this one.
Question.
Yes, Andy?
In that last formula, what have you done? What is K? You said
Okay, I'm jumping too much. K is the motility tensor. It's what I call the motility tensor. So from the formula, K is on the other side. Okay. So the third equation might be rewritten like this: 1K divergence of TC plus MCA. Okay.
Why is it the same TC appearing with the T in the velocity and the divergence? Is it
Because it's this one. It's just I'm just writing this one.
no, because
Okay.
you have VC is also
Oh, sorry. No, VC. Sorry, this is VC.
but VC contain also the divergence.
VC contains the divergence. This is not a plus. This is a positive part of this one because it comes from this one.
Is that the same equation, or you're saying the
No, this equation here. Put in the K.
Yeah.
Put in the K. I'm sorry. This is equal VC because there is a minus sign. Okay. If I put this one, just this here, K minus 1 goes to the right hand side, then I put K on the left hand side and I get this one. Okay, this is usual. Now I'm saying this motility, I call it motility tensor, which is the tensor structure depends on the fact that in some tissues like the brain, you have fibers aligned in certain direction and you want to keep track of this anisotropy. And instead here I'm writing that as a scalar in isotropic situations. And so I'm saying, okay, this K, this motility vanishes below a certain A naught.
But that won't reflect the bimodality, will it?
No.
This just
In
captures the
fact, I'm saying if you want to match this one, then this is enough. Okay. Jimmy, you had a question?
Yes, somewhat related. So I see this equation, I see this is inspired by Darcy's law, right?
Yeah.
You would see K as the permeability.
Yeah.
Darcy's law is purely viscous, it's purely dissipative. And now the entry of the nucleus or the cell itself, there's an elastic component to it. Wouldn't that require a fundamental change in the character of this force law?
Well, at the moment, this one is just general mixture theory. You can do whatever you want, but I mean, this is basic. The assumption that you are making on to get Darcy's law is more or less related to how you treat this TC and how you treat this K here. I mean, if you have an elastic body instead of a viscous force, then you play with this TC here.
But what I'm saying is the force may not be proportional to the instantaneous velocity, but rather
Yeah,
the
sure.
strain would have
No. In fact, I agree with that. This is just if you assume that is proportional to the velocity, you can write this one. If it is not, you have a more complex law which might not be easily invertible. Okay, because you have some force that is a nonlinear dependence on the velocity. So.
Well, that last equation, I'm still confused. I mean, you have a vector on one side, and then you have a scalar. So it's written in one dimension. Is that what you mean?
No, which one? This one?
Yes, that one.
This one is a vector, this is a scalar, this is the divergence of a tensor.
Yeah, so I don't like a vector equals a scale. Can you explain?
It was a prefactor.
It's a prefactor.
It's a prefactor.
Oh, it's the plus sign.
This plus here is the positive part.
Yeah, sorry.
I already had this question.
Yeah.
I was prepared. This is a positive part of this parenthesis here. It's not a plus.
I mean, concerning the comment about Darcy's law, another way to understand it is that all you're doing is you're defining a term for the momentum transfer between the two phases, but you're not worried about the phase that's receiving the momentum.
Yeah.
Correct?
Yeah. Okay, so going a step ahead, a way to put some nonlinearity and to say bimodal behavior that is continuing this curve and have either a saturation, that is the velocity saturated at some point, or say a maximum and then a minimum, is to put a denominator here. Okay. Unfortunately, I have no idea on how to measure this A1 because there are no experiments. We could infer that from the cellular post model, but I mean, it's not really an experiment. Okay. Okay, so this is what you have. Now, the other thing is that A0 is not a constant, in the sense that this threshold value, which is unique in some sense in the experiments by Katerina Wolf and Peter Friedel, as we learned, depends a lot on other things, the nucleus size, of course, but also on the elasticity of the nucleus. And they show that according to the elasticity, you have cells that can squeeze on the same pore size and others that cannot. Okay, so this is not really constant. It is as a function. It's a function of a lot of things, and we can infer that from this curve here. Okay, this is what it says. I mean, this says that the A0 here, the radius of the pore, is related with the areas of the pores, of course, is related to some balance between, say, the forces here at the bottom and nucleus elasticity. Okay, just reverse this function. You have something like this, and then it tells you how A0 depends on this ratio of the forces with respect to the stiffness. Okay. Well, we can use some specialize this growth term here, this right-hand side for the growth of the cells, taking into account of contact inhibition of growth, remodeling of the ECM by the cells, production of metalloproteinases in some of the cases that I will show. So, as I mentioned at the beginning, my aim was to find a model that in which cell can be segregated by a porous structure like a basal membrane, which was not possible to be done by previous model because, of course, the porous membrane is always porous and the cells are treated as a fluid and they can always pass through this membrane. What happened? Okay.
Your computer's
Oh, come on. You want? No, I think that he wants to close and. Okay, update some software. And the right timing.
16%, 17%.
Okay. Well, I will try to continue at least for some time. So the idea is this one, that you have this porous wall and you want the cell to penetrate penetrate or not according to the properties of this dense region of ECM. Okay, and unfortunately I had now the graphs, and so I cannot describe the graphs.
Question while you're waiting. Do you have any idea in Peter Friedel's movie what the relative forces those different cell types can exert? Do they have any data on that? Well, it's not clear that it's specifically the nuclear properties that's restricting the motion. It could also be that one cell type can't exert much force.
I see that they are not measuring the forces, but now they are measuring the stiffness of the nucleus. They are doing that. I'm not really sure whether they published this experiment on. They're poking on the nucleus to find the constitutive relationship of the nucleus, so we can get some answer.
I didn't quite see how contact inhibition was coded in the continuum model. Could you once?
Oh, it's just very easy in the sense that when the volume ratio reaches a certain threshold, you switch off growth.
Oh, okay. It's not really contact then.
Well, no, if you want, it's pressure inhibition, but it's not even pressure, it's just volume ratio. Really.
Limit on the volume.
Yeah, it's a limit on the volume. Yeah, Jason?
One question on the definition of BC. So actually, it's the velocity of deformation of the cell seen as a geometrical object, not the velocity of the actin, for instance.
No. It's a macroscopic quantity, because you have to take into account that this is a macroscopic model, so it's a velocity of the ensemble of cells, actually.
Okay, so which means that your the friction force is the opposite sign from Alex Mogilner's velocity, because if it's the velocity of the protruding cell, so the velocity is forward, which means that the friction, the resistance is going in the opposite direction. Whereas if you have the retrograde flow and the retrograde flow is hindered by
no, I'm not taking into account any retrograde flow or something like this.
So your forces would be the other way around compared to.
Yeah, it's in direction. At the end of the story, this has to be into direction of motion. This one. When you want to.
Ooh, perfect time. Well, you mentioned a moment ago that you can think of this as an ensemble of cells, right?
Yeah, more or less in continuous, you're looking at more.
But, are you thinking of a connected region of cells moving collectively through? Or
more or less an aggregate of cells. At the end of the story, what I want to describe is an aggregate of cells.
But then again, the cells in the interior of that aggregate aren't going to be in contact with the ECM.
Yeah, this is true. Well, partly true, because you have a CM also inside the aggregate. Sorry about that.
Are you going to show us the constitutive relation for the ECM pore size, where it's increased by MMPs and it decreases because there's spontaneous polymerization?
No, what I'm going to do just is in one of the simulations, assuming that the MMP is degraded by the cells and produced by the cells at a certain rate, and so with this will change a bit the density of the cells and then the pore size.
And you're going to keep it isotropic, the pore size?
Isotropic, and in order to relate, say, the density of the ECM to the pore size, I'm also assuming that this is regular. Otherwise, I don't know how to do that.
Well, we could, in principle, extend it with some model where you have spontaneous polymerization of collagen, for example, and breakdown, and you could have it make the pore size a tensor as well.
Yes. Okay, hopefully it will start again.
May I have one technical question?
Yeah.
I just wanted to ask, you talked about fiber elasticity. In these experiments, this is all in collagen metric.
Yeah.
And do you know how the elasticity is modified?
It's just you modify in a zero post model. You modify, say, a certain parameter that is related to the elasticity of the cell.
So I thought like experimentally, or this is just from...
No, no, in this one I'm doing that just from, say from the simulation point of view.
You have another question, Luigi?
Yeah. Well.
I also have a question. So, what is the difference between bovine and red matrices?
The fibril of the bovine is 60 nanometers, so it's thicker. The one of the rat tail is 20 nanometers. Say, from scratch, say bovine collagen have wider, say, pores. Then you have to also take into account that these fibrils, in some sense, merge and form thicker fibers. But from the stretch, I mean, this makes a difference.
Any correlation between nuclear stiffness and transfer?
Is there a relationship?
Yeah.
I don't know if there is a biological relationship between the two. But I don't, if there is a relationship between nuclear stiffness and forces. What I know, for instance, is that in the experiments that they are doing, because they are working with the Frieder, they are interfering with laminin in some sense. Okay, yep.
Dennis Discher has published quite a bit on that, and they measured the nuclear stiffness as a function of the lamine composition and could also tune it by knockdown. And it turned out that the softer the nucleus, the easier the cells can penetrate through pores. But the downside is if the nucleus was deformed too much, these cells would die a while afterwards. For that cell type. I'm not sure if this is universal.
So this is the first simple simulation. You have some cells living in an environment with 20% of ECM. They grow, but they bump into this region with a higher density of ECM, and they stop. They just stop there. They will not invade. There is no diffusion, no degradation in this case, and stay there forever. If this 30% is lowered to 25%, then instead this value is not critical, it's supercritical in some sense, and they can go through and invade also this area with a higher stiffness. And this higher, volume ratio, a lower pore dimension, and this is what we wanted. And well, here is what I mentioned about the MMP. Instead, if you have metalloproteinases with the 30% region of ECM, no problem. Just the cell will just degrade ECM and continue growing. And this is the same thing in two dimension. You have in some sense a barrier here with 30% of ECM. The cells are growing in an environment that is full of normal cells. These are tumor cells in some sense. These are normal cells here, and here it's not in the picture. Below and above the membrane, in some sense, this thicker region. In one case, the cell will just go on and continue moving like in a duct, like in a ductal carcinoma or whatever. And in this one, instead, they will invade this region, pass through this denser region of ECM, and invade the outside. And of course, the normal tissue will just die because of compressed by the tumor cells and so on. Okay, this is more important. This is a comparison with what happens when you have a stiffer nucleus and a softer nucleus. The nucleus is here. This changes a number in the simulation, and this is in some sense a cut of this simulation, a vertical cut. You see the membrane here. The blue cells are the normal cells. The red cells are these tumor cells that are softer. When they reach the membrane, the blue cells are not present into this area here, and they will not be present there. They are present in and out. But when the red cell reaches, the red cells reach this area, they are able to invade this ECM and then invade also the outside of the tissue. Just because the nucleus is stiff, is softer, they can pass through that. And of course, you can play with that even in a very heterogeneous environment. You might have regions with different ECM density, and they are circumvented in some sense by the self, and a region where the cell can, the tissue can pass through. Okay, this is just a toy model toy numerical simulation to see that things work. Okay, now in the little time that was left by my computer, I would like just to go a little bit in details on the first, how to relate then now the microscopic, say some macroscopic understanding of cell traction to macroscopic parameter gain. And the idea then is the following. So now I'm focusing on the force as the one that are causing this interaction force between the cell and the extracellular matrix. In principle, what you have, I think we have learned that very well. Cells are binding to the extracellular substratum with some bonds, and these bonds are stretched, some are broken, some will form on the tip, and so on. And this, from the mechanical point of view, means the following: that you have two continua, say the cell and the substratum. Say this one, the white one is the cell, and this one is the substratum. And at some point of their history, there is a bond that is forming at some point, and this bond then, because the two constituents are moving apart, this bond is stretched, and so this will, for instance, exert a force on the ECM if this is stretched of this amount alpha beta. Okay. So the idea is then that this interaction force between the cell and the extracellular matrix is the sum of all the macroscopic forces that are acting below the cells. Actually, to tell you the truth, as I'm treating in the continuum, I would say the ensemble of cells, in the sense that the point here, this one, this XC is big. Okay, in order to have to be possible to say treat that as a continuum. So it's just the sum. This is the sum of all the contribution, and these contributions here depends on this distance here, in the sense that you have a bond that is formed at some point when the two are found in the same place, and then this bond is stretched, and this will generate a force. Of course, this is a passive way of treating things. And okay, so in order to do that, what I will introduce is a distribution function of the age of the bond, okay, so that this integral can be written as an integral over this distribution function, which means I'm summing the forces that are exerted by the bonds that have a certain age. Because, for instance, the bonds that are formed now, probably they are not stretched too much. Those that are older might be stretched a lot. Okay, and this n is the total number of bonds. The total number of bonds, and of course you need an evolution equation for that, for this distribution function, which is this one, that tells you the following. It tells you that, well, if you have a distribution of bonds, and time is going by, then these bonds are just aging. Okay, this is this operator here. That is, after 1 second, they are 1 second older. But while they are aging, they may rupture. Okay, and so this is the detraction rate of the bonds. And of course, they can form. New bonds can form with the age equals 0. So the forming of the bonds, the generation of bonds are related to some boundary condition for this equation here. And I'm assuming that this is proportional, at a certain rate, is proportional to the say the difference between the maximum number of integrins that are available to the number of bonds that are already active, okay? And so I can play with this equation. Okay, luckily, bonds are renewing very fast compared to the, say, the velocity, the typical velocity of the cells. So the bond age is much smaller than the travel time, or equivalent, one could prove that this is equivalent to saying that the breaking length of the bond is much smaller than the cell diameter. I think that this can be understood. I mean, you cannot have a bond that is 10 microns long, okay? So if you do that, this means that one can drop this time derivative and ends up with an equation that is, in some sense, is solvable. I don't want to say that it's easy to solve, but I mean, it's more or less an ODE. It can be done. So this gives you the distribution of bonds in a quasi-static situation. Forget about the formula, just take the principle of that. I mean, since the bonds are renewing, there is a distribution of bonds that are acting below this cells or ensemble of cells, and this distribution depends on what? On how fast the bonds are formed, on the maximum number of catenins of the of addition bonds, on the degradation rates of the bonds, and this one is a function of the microscopic force. Okay. So what you see here, these are all quantities that say live in a subcellular or cellular space, in some sense, at that level of description. Yes?
How does this formula account for the fact that there are different bonds with different lifetimes and different forces?
Well, if you do that, you are making the model more complex, and you have to have more, say, distribution function and so on. Okay, because you have say more population of bonds, for instance.
This is the mean for all bonds.
Yeah, some sense like that. Okay. Okay. So, but the important thing is the following: that since the force, as I told you, is just an integral of this of the microscopic force by this distribution function, you can, in some sense, have an idea on the shape of this macroscopic force on something that is related to microscopic parameters. Okay, so coming back to the title of the talk, if this is multiscale or not, I don't know. And actually, to tell you the truth, to answer a basic question posed by Hans Ottmer, well, I'm not too clever, but I don't see that there is a single way to say upscale the information and do upscaling. It depends on the problem. Okay, so is this one? I'm not sure. I mean, and I'm not sure that you can generalize that one. This is working for this case, in my opinion, but probably it will not work for all cases. But it's certainly a way of relating this interaction, this macroscopic parameter, with something that occurs here at the microscopic level. So let's play a bit on these terms here to try to understand what is the meaning of the laws that are already described in the talk. For instance, okay, well, of course, I'm assuming some kind of Hooke's law. This simplifies a little bit things. Forget about that. But okay, let's assume that the bonds at the membrane are just renewing all the time. Okay, constantly renewing at a certain rate, regardless of the force that is acting on the bond. You put that into this formula here. This is just a constant. You integrate it, and what you get? You get the interaction force is proportional to the relative velocity. Okay? So what I mean is that when one is assuming that the interaction force between the cell and the extracellular matrix is just linearly proportional to the relative velocity of the cell with respect to the extracellular matrix, in some sense it's assuming at the microscopic level that the bonds are continually reformed. There is a trafficking of the integrin at the membrane, and bonds are broken all the time, and renewed all the time. Okay, which is probably true in embryogenesis, for instance, is very likely to be like that. But for more mature tissues, probably this is not so true. Instead, if you allow a threshold in order to break up the bond, that is, the bonds break up only if there is a minimal force that is acting on that. You again play with the formulas, and what you get is something like this one. That is, you don't start from the origin, but you have a threshold value of the interaction force, macroscopic value of the interaction force, and from that you have the relative velocity that goes in some sense up, the increase with the relative velocity, which means that in order to have a relative motion, the aggregate has to be to feel a minimal force, a minimal stress, and then it will move with respect to the extracellular matrix. Otherwise, it will stick to the extracellular matrix. Okay, this is what I'm saying. If cells are not pushed, or actively pulling strong enough, they will stick to the extracellular matrix. Otherwise, they will move relative to the extracellular matrix. Okay, this is a sort of yield stress in continuum mechanics. But we can do a bit more, because in some sense we know the strength of the bond as a function of the force of the unbinding force. These are some experiments that are done, for instance, by Sun and by Baumgartner, and there are a list of papers that do that with the atomic force microscopy. They just put the bead in contact with the cell and then they pull in order to measure when the bond will break up. Okay, and so here you have, say, the number of events, if you want the probability of an event, according to the binding force. Okay, and you can relate that to this decay rate of the bonds to a survival probability. It is a function like this one, which tells you that there is no breaking below, say, 7 piconewton. Then the breaking rate is increasing, and there is a maximum rate, say, around, I don't know, 80 piconewton, meaning that at this force, 80 piconewton, all the bonds are broken. There is no chance to have a bond that is surviving. Okay, you can put that into the formula again, and you have a graph like this one. Okay, you have a threshold value, then a maximum, and then a decrease. And whether this one can be related to one that Alex Musgilner has shown yesterday, I'm not really sure, but I mean, you have this behavior like this. Okay, traction force, then a decay. Of course, here you have the acting flow, and here you have the relative velocity of the cell with respect to the extracellular matrix. To tell you the truth, this is bad. This decreasing function is bad, because from the point of view from the macroscopic point of view, this is an unstable branch. Okay, like in phase transition. But in 3 dimension, likely you have another contribution which is related to the fact that the cell has to find its way through this network, and this brings up an extra contribution to this force that is that makes things go like this one. Okay, so this is the unstable branch, and this is a stable branch, which means, for instance, that again, in order to have some relative velocity, you need to overcome some threshold. Then it will move slowly. If you the force is too large, then you jump to this upper manifold, and so on. Okay. Or from the other point of view, if you allow the cell to change this threshold value, okay, you can have situation in which, assume that this one is the force that is exerted on this aggregate, on this specimen, these cells that are over here will stay still, they will not move, but this one, the green one, will move with respect to the extracellular matrix. That is, changing the properties of what? of this threshold value that is related to the minimum force that is needed to detach the bond. If this one is lowered, this number is lowered, then this one is lowered, and then you have, say, the difference between staying still in an extracellular matrix or move with respect to it. Okay. Okay, so I'm not really sure. I had another topic, but probably I will leave that. But for those who are interested, this has to do with traction force microscopy, that we were doing something on 2 dimensions, and now we have an algorithm to do that in 3 dimensions. And the thing is a bit different, in the sense that in 3 dimensions, in 2 dimensions, what we have in traction force microscopy is that one has measurements all over the regions, and one knows that the forces are exerted below the cells, okay, which is part of the domain. Instead, in 3D, we have something different in the sense that the measurements are done outside the cells. There are no beads inside the cell. And the traction forces are exerted on the boundary of the cell. So the domain is this omega with a hole, and the measurements are in omega in the hole, and what you want to infer is the forces that are exerted on the boundary. Okay, this makes a little difference in the algorithm, in the inverse problem, but this can be done, and so we don't have data yet, but if you have data, we are happy to play with that and try to infer in some sense from the deformation here, from this field of deformation, the forces that are exerted on the cell, by the cell. Okay, these are a simulation is obtained in this way. We are imposing this force. We are deducing from that the deformation of the gel. Then we are going, say, in the other way, imposing this deformation. We are trying to see whether we can recover the one that we imposed, and we are not too far. So, I mean, the algorithm should work quite well. But, I mean, this is just for advertisement. And thanks for your attention. Yeah.
So, you showed this nice graph where you related geometry to the force and stiffness of the cell. But I also mentioned that degradation of the extracellular matrix is an important factor. So I wonder if in your model you can continuously change the degradation rate, or sort of somehow some measure of the degradation, and see if there are interesting, thresholds or transitions happen based on that. Because I assume you're considering extreme cases where you have no degradation or a lot of degradation. But like in the disease state, you might have it's affected to some degree, but not fully. Have you ever tried to?
Well, the degradation, the part in the model that is devoted to degradation is in some sense a different story that will give you the structure of the ECM, okay, and so it will pass through or not. In the sense that assume that degradation is not 0 but is small. Of course, the cell will not pass through at first, and then will start passing through very slowly when the channel widens up. So, in some sense, we are able to take into account, say, the timing, how much the degradation is going on, and the remodeling by, I don't know, other things like fibroblasts or whatever.
You can, but you guys didn't do this, right?
Yeah, you can do that.
It's easy to incorporate in the extracellular matrix a nonlinear behavior of the ECM.
Yes, in the sense that in the simulation we show the matrix is rigid, but as soon as you want to have it deformable, you just put a constitutive equation for the stress, whatever you like, and you have a deformable extracellular matrix. Yes.
The image on the screen shows the cell in 3D matrix,
and it shows all the force going inside the cell. Is this possible? Mustn't there be an equilibrium?
Yeah. No. This is
forcing showing.
No. The argument is done so that there is equilibrium. In the sense, the space of forces is such that the resultant and the momentum is 0.
The arrows show.
Yeah, and so the resultant is 0.
But shouldn't contract the cell under these conditions?
Well, probably in this situation, I don't know, because it is attached to the extracellular matrix. I'm not really sure. But if the extracellular matrix is deformable, we will contract. We pull the ECM. This is just a toy, just to see if the algorithm works. But of course, if the ECM is deformable here and here, it will pull the ECM in this direction. I mean, it's just in 2D when you are wrapping the substrate, more or less. But then you are in 3D now. I'm not saying that this is just what's happening. We are imposing the figure on the top. We are measuring the formation. We are putting that one as an input of the inverse problem, and we are recovering this one. So one is just to compare the top and the bottom to see the difference.
Do we have any other questions? So I think we are at exactly 10:15, which is like a very good time. So let us thank the speaker again.
Okay.